Hello, myself Dr. Prasad Puthiyillam. Welcome back to another class on nanotechnology, devices and application, nanoparticle for sensor and circuitry. This particular class video covers the topics such as photo-induced electron transport in DNA, electronic devices based on DNA, photo-induced charge transport, electrical manipulation of DNA on metal surface, DNA gold nanoconjugate, non-invasive biosensor in clinical analysis, applications of biosensor-based instrument to the bioprocess industry, biosensor for environmental sample detection, biochip or biosensor, Biacore, and optical biosensor. The class videos are available in my YouTube channel, My Intuition. First topic, that is photo-induced electron transport in DNA. Okay, so to know about DNA, the structure of DNA is given, the compositional structure of DNA is given here. We know that DNA consists of mainly 4 bases: adenine, thymine, cytosine, and guanine, and consists of a backbone made up of phosphorylated sugar. The DNA or deoxyribonucleic acid is one of the very famous, most famous biopolymer, and it is very unique. It's a 3-dimensional molecule, and it carries the genetic information of all living organisms. DNA can exist in 3 different conformations, including A, B, and Z form, in which B form is the most common. Now the question of whether DNA conduct electricity or it can conduct electric charges or not, it is an interesting research topic to all fields of science and technology. The continuing progress of nanotechnologies and the consequent need for further size miniaturization makes DNA molecule an excellent candidate for molecular electronics. DNA might serve as a wire or transistor or switch or rectifier depending upon its electronic properties. The experiments which are conducted based on DNA electron transport, DNA usually it available, or its natural availability in the form of liquid solution. Therefore, experiment can be conducted in the solution state, or even artificially imposed dry environment can be created to study the DNA. In dry DNA, the experiments on dry DNA, direct conductivity measurements can be done on DNA, already done on DNA, but give controversial results. Like some say that DNA is a molecular wire, it's a semiconductor, or it is an insulator. So these controversial results are due to the fact that they are not done the experimental in a well-controlled way, or still we are lacking that experimental control over studies on DNA. So this includes the experimental control required, methods for DNA alignment and drying, the nature of devices used to measure the conductivity, or the type of metallic contacts used, as well as the sequence and length of the DNA which is used. In solution state, comparatively easy to do the experiment. The DNA chemically composed or processed to host a donor as well as an acceptor molecule at different sites along its long axis, and then photo-induced charge transfer rate can be measured. Based on this experiment and results, it is possible to obtain a very clear picture of the charge transport phenomenon in DNA. And now it is clear that DNA-mediated charge transport process occur in a very fast or even ultra-fast time scales. The knowledge about this electron transport in DNA, it has got a potential to be considered for its development as DNA-based electronic devices. The electron transport in DNA can be used to develop electrochemical DNA chips, or even for the detection of single base mutations. Electronic devices based on DNA. To study about electronic devices based on DNA, we should know about the solubility of DNA. So DNA, it is actually a negatively charged polyelectrolyte, which is covered by positively charged inorganic cations like sodium ions. The purified DNA usually is soluble in water, therefore, or it is insoluble in organic solvent, which restricts its applications. Therefore, effort has been done to make DNA soluble in DNA by using surfactants, or surfactants are used to improve the solubility of DNA in organic solvents. The different cationic surfactants are used, which includes cetyl trimethyl ammonium chloride, CTMA, octadecyl trimethyl ammonium chloride, OTMA or lauryl chloride LAU are used in preparation of DNA-based complexes which are soluble in organic solvent. These complexes can be easily fabricated by using spin coating or dip casting or printing techniques. The film of DNA with CTMA complex has been employed in a laser and as a non-linear optical material. Now, the application of DNA as a functional layer in organic thin film transistor or OTFT. The basic structure of OTFT consists of a three functional layer, an insulating layer, which also we call as gate electric, a semiconducting organic layer of P-type or N-type or even MP polar, and three electrodes, three conducting electrodes like source, drain, and gate electrodes are used. Now, recently, the base pairs of DNA also been studied as the gate insulator in organic thin film transistor. Among the base pair, the adenine guanine base pair is found to have a higher dielectric constant and a lower dielectric loss, and they break down strengths, which are a promising factor for being a dielectric for OTFT. Now, the other electronic applications of DNA, if you discuss, other than OTFT, the bioorganic polymers from DNA has been used to prepare photo-induced write one, read many times memory devices. Memristive memory devices are the other application than that of OTFT. Even the DNA and other nucleic acid bases were explored as a hole blocking layer or effective electron blocking layer in bio-LEDs, which effectively enhanced the blue and green light emission. Next one, photo-induced charge transfer. Photo-induced transfer studies are done by posting a donor as well as acceptor molecule in the different sites of the DNA long chain. The requirement for the photo-induced transfer rates are the different sites of donor and acceptor molecules, or the distance between the sequences of DNA that lies between them also affect the photo-induced transfer studies. In order to do this study, the requirement, it is crucial to modify the structure of DNA with suitable chromophores. Chromophores are the molecule which gives color, and to prepare for the compounds which gives color, and the well-defined DNA structure also required. It should have a pi system, alternate pi bonds required, or conjugation system is required to do the conductivity studies. Now it has got two types, that is photo-induced oxidative hole transport or photo-induced reductive electron transport. This both cases, first case, that is hole transport, that is means positive charge centers are created by oxidative process. These positive charges can be transported in the opposite direction, electrons will be moving. Or the reductive electron transport, anyhow it is the electron which is transported. So both processes, it is an electron migration reaction, but with a different orbital control, molecular orbital control. The oxidative hole transport, it is supported by the highest occupied molecular orbital, whereas the electron transport, it is supported by the lowest unoccupied molecular orbital controlled. That means the electron movement takes place in this particular molecular orbital. Now, the electrical manipulation of DNA on metal surfaces. Electrical manipulation electrical manipulation studies on metal surfaces are having very much importance, because it will give the physical supporting the physical science or molecular biology or even nanobiotechnology. The electrical manipulation of DNA on metal surfaces carry much importance. DNA metal surface interactions, the interaction between DNA and metal surface are not covalent bonded. They will not form any covalent bond formation. It depends upon the mechanism, like a molecular backbone or a basis interaction with the metal, or electrostatic interaction, or van der Waals interactions among the DNA and metal surface. Now, the immobilization strategies. Immobilization means how we make the DNA to immobilize on the metal surface. The DNA, once it is attached to the metal surface, it should not move from the surface. Then the studies are not possible. We call it as immobilization. The strategies which are used the immobilization function of DNA layers on supporting metal surfaces is very important to study the electrical properties of DNA and its application. In the field of self-assembled monolayer, that is SAMs, the widely used method employs the thiol-terminated molecular linker group to bind to a metal surface through or via a sulfur metal bond, which is assisted by van der Waals forces and hydrophobic interactions. Now, after attaching the DNA molecule on the metal surface, the different characterizations were done as the surface structure and coverage by radio labeling or neutron reflection or X-ray photoelectron spectroscopy (XPS) or even IR spectroscopy. The molecular structural studies of nucleic acid layers bound to the metal surface were done with scanning tunneling microscopy, atomic force microscopy, and surface plasma resonance. Next topic, that is DNA gold nanoconjugate. That is DNA attached to the gold called as DNA gold nanoconjugate. DNA functionalized gold nanoparticles are an interesting system with application ranging from biological sensor to the construction of a self-assembled material. Different experiments were conducted by attaching a single-stranded DNA molecule through the thiol bond to the surface of gold nanoparticle, and the subsequent self-assembly process or the conjugate by making use of base pairing of complementary DNA molecules are studied. Due to the strong interaction between the gold and thiol, a single monolayer of DNA around the gold nanoparticle can be obtained. Even the negative charge repulsion of the phosphate backbone of DNA, which orients the DNA strands out into the solution with a unique footprint, it depends upon the particle size of gold as well as its packing density. Next topic, that is non-invasive biosensor in clinical analysis. The non-invasive methods, what we say, they are the procedure which are strictly defined without breaking the skin or no break on the skin is created. There is no contact with the mucosa or the internal body parts. The interaction is through the cavity, the natural cavity present on the skin, or any artificial body orifice are used for that. The use of non-invasive biosensor for health monitoring, it is a very new approach. For example, the IR light sensor used to detect glucose level without puncturing the skin, which passes the IR light on the skin, and the absorption rate of IR by the glucose or the sugar, it is measured, and based on that, the detection is done. Now, non-invasive blood glucose sensor, it uses usually electromagnetic waves are used to sense the blood glucose level by changing the radio wave impedance using the spectroscopy. Varying the radio wave frequencies, the changes in blood glucose is determined. But in such cases, the calibration of the instrument is a little bit problematic, difficult work for the people who are handling that. Then iontophoresis is a new technique which which is introduced ionic medicinal compound, which induces ionic medicinal compounds into the body through the skin by applying a small current or electric current to the skin. This process causes increased molecular transport through the skin and has found applications in transdermal drug delivery. Such a schematic representation of the device it is given there. Now, applications of biosensor-based instrument to the bioprocess industry. Bioprocessing, it is the use of biological material. Usually, enzymes are used for the production or commercial production of different products. Okay, so by using enzymes, their importance, when we say the importance of bioprocessing, they are too specific and produces a very pure product. They are very efficient because very little amount of enzyme can be used as a catalyst to produce a large lot of product, and they are biodegradable, causes a very less environmental pollution. Therefore, the advantages of bioprocessing are that it requires very low temperature, low pressure, low pH. They can be used they can use renewable resources as a raw material. Large amount of products can be obtained with very less energy consumption, and the process can be scaled and possess economic viability. Even it is able to be able to separate the intermediates and end product from the biomass stream. A typical bioreactor, it is represented here. A bioreactor is a vessel used for the bioprocessing. It is a chemical process which is done with the help of enzymes. It consists of system monitor, system props, thermal jackets, submerged aerator, the reaction tank, and the agitation system through which the air and the medium are pumped and made to react. Now the applications of biosensor based instrument to the bioprocessing, it is used for monitoring the growth and changes of microorganisms which will badly contaminate the product. Used for monitoring the actions of enzymes which are held in suspension with the nutrient medium, for monitoring the temperature or pH or the substrate and product concentration. So bioprocess industries is a new industry, new approach of production of a large scale amount of product, to pure product without harming the environment. There we need to have a biosensor based instrument for monitoring different functions. Next, biosensor for environmental sample detection or polluting element detection. The main polluting or sample or the elements which pollute or treated as pollutants are heavy metals, organic pollutants, nitrogen compounds, polychlorinated biphenyls or phenolic compounds. We'll discuss one by one. Heavy metals. Heavy metals, even a very small concentration, if it is present in the environment, causes human health issues, and they are non-biodegradable also. So many of the bacteria biosensors developed for analysis of heavy metals in environmental samples make use of a specific gene responsible for bacteria resistance to these elements, such as biological receptors. Now, these bacteria strains resistant to some metals such as zinc, copper, silver, tin, cobalt, or mercury, they have been isolated as possible biological receptors. Now, organic pollutants. The organic pollutants we measure in terms of biological oxygen demand, BOD. That is a parameter which is used to indicate the amount of biodegradable organic material present in water. If these biodegradable organic materials are present in water, they will make use of the dissolved oxygen for their degradation, and which causes the dissolved oxygen concentration deficiency in water bodies, which causes the aquatic organism. Okay, so this is done. The fast determination of BOD need to be done. It is achieved with the biosensor-based methods, in which the bacteria respiration rate, that is, say, example with E. coli, in close proximity to the transducer, it is measured. With this method of determination of BOD, a real-time analysis of multiple samples are also possible, and they are handy devices which are available in market right nowadays. Now, the next one, nitrogen compounds. Nitrates are widely used to. Nitrates are widely used for food preservation and fertilization of soil. But continuous consumption of nitrates can cause serious health issues which will affect our hemoglobin count. So the biosensor based on cytochrome C nitrite reductase, that is CCNIR, it can be used for the determination of nitrate by measuring the electric current or changes in electric current. And these are very fast sensing. The response is very fast, within a 5 seconds or so. It will give the linear changes in the as a response. Then conductometric electrodes, which are modified with methyl vialogens. Vialogens are the molecule which have two positive charge centers in a molecule. These are used as a mediator mixed with nitrate reductase enzymes as a biosensor and used for the detection of nitrate. So nitrate reductase enzymes are already available in natural. So making use of that, conductometric electrodes are prepared, and it is possible to measure, or it is done the nitrate determination, qualitative as well as quantitative determination is there. Next one, that is polychlorinated biphenyls. These are toxic organic compounds which are treated as an environmental pollutant. The newer technology introduced, like immunosensor technology, are used for the determination of polychlorinated biphenyls. These immunosensor are class of biosensors, which uses recognition elements like antibodies and antigens. Next to that is phenolic compounds. Phenols are derivatives, and their phenol and their derivatives possess high toxicity. They are produced or polluting through industrial effluents, or due to the production of or the industries like plastic, polymer industries, drug industries, dye industries, pesticide industries, detergent industries, oil refinery industries, or pulp and paper industries causes phenolic compound pollution. The toxic pollutants in wastewater usually interact with the DNA also, which leads to the damage in human DNA. These interactions of DNA with this pollutant are only made use to make it as a sensor. Okay, so the interaction between the toxic pollutants and DNA is used to prepare electrochemical DNA biosensors. Next one, that is organophosphorus compounds. Organophosphorus compounds are a group of chemicals which are widely used as insecticides or herbicides or dioxins. Some of the biomolecule like peroxidase, alkaline phosphate are used to detect pesticides. Biomolecule like acetylchlorinesterases, cyanobacteria are used in biosensor for the detection of herbicides. And dioxins, they are the polychlorinated dibenzopyradioxins and dibenzofurans. They are the chemical compounds collectively called as dioxins. These dioxins are potentially toxic substance for human, and causes the impact on environment and reach the food chain accidentally. They'll be available in water as well as soil as well as the detection of dioxins are done with biosensor based on enzymes or antibodies, DNA, whole cell, and other biological receptors are used. Next one, biochip or the biosensor. Biochip, when we say, they are the microelectronic devices or microelectronic inspired devices which are used for delivery or processing or analysis or even detection of biological molecules and different biological species. These devices are used to detect cells, microorganisms, viruses, proteins, DNA, and related nucleic acids, so small molecules are detected by using biochips. Some of the commercially available biochips are Nanogen or Affimetrin and Caliper. The advantages of using biochip that they have a small length scale, they require small thermal masses, and they have a high surface to volume ratio. The last topic, that is Biacore, an industry or industry which produces an optical biosensor. They are revolutionized the scientific research by enabling a real-time detection and monitoring of biomolecular binding events for a better understanding of biochemical reaction mechanisms. Biacore, they specialize in measuring biomolecular interactions, which include protein-protein or small molecule or small fragments with protein interactions. Its technology, it is often used to measure the binding affinities as well as kinetic rate constant, even thermodynamics of that. The targeted molecule or the analyte, their binding interaction with the sensor molecule on the sensor chip surface is used for the quantitative measurements. And the interaction between proteins, nucleic acid, lipids, carbohydrates, and even whole cells can be studied by using Biacore. The typical schematic representation of a Biacore is given here, in which the sensor chip consists of a glass surface coated with a thin layer of gold. The most widely used sensor chip for the gold surface, it is modified with the carboxy methylated dextran layer. So a dextran layer is given there. This dextran hydrogel layer forms a hydrophilic environment for the attached molecule, preserving them in non-denatured state. That means analyte will go and attach on this dextran, which causes the change in the current or detection is possible, and it will not affect the analyte also. So that about the topic that is nano particle for sensing and circuitry, as well as some of the nanobiological sensor devices. The class is available in my YouTube channel, My Intuition. I hope that I gave some information for you. So please do like my videos, share to maximum. So thank you very much for watching my video class.